Thank you for that overly kind introduction. It is, and Jeff and Chuck, both of you, thank you for the invitation. It's particularly satisfying for me to give this talk to my BME colleagues at your behest, Chuck, because as you said, when you were running the Culture Award, you really helped catalyze a lot of the success to date. I mean, we've got a long way to go, but what we've seen so far with the startup you mentioned, LifeSprout, and I will be telling a little bit of that story today. You know, that Culture Award was the first money in, and it was a pivotal translational award for Haichuan and myself to get all this started. At that point, that 100,000 is now translated into more than 30 million that we've raised. We hadn't even made that product outside the lab. We've now done GMP manufacturing, and the first products are in clinical trials. So again, I'm very grateful for the faith that you had those years ago, and hopefully eventually this will turn into a product and something very valuable for everybody. So I'm going to tell you today about a very broad theme, commercializing your discoveries. Or I think you had asked me, Chuck, to talk about how you evaluate technologies to see if they're ready for commercialization. And you had mentioned, a couple of the things that I do. One of my most enjoyable, probably my favorite job at Hopkins, of the few things that I do, is to be the senior medical director for Johns Hopkins Technology Ventures. And there I work with a terrific group of colleagues on the senior team: Christy Weiskell, Brian Stansky, Steve Kosouris, many others, and throughout the organization to help really do two things. I work with some of our most prominent faculty and some of the best technologies we have in the university, and try to develop commercialization strategies, so to figure out how to cultivate the real world import of those discoveries. And that's on a one-on-one basis. It's a little bit tactical. The other half of what I do is more strategic. It's working with the aforementioned leaders within JHTV, as well as leaders within the university. And in fact, Jeff is one of those. He's on the faculty advisory committee with me for JHTV. And we all think about how to reduce the friction involved in technology development. And it's some of those learnings that I want to share with you today. I'm going to try to do this, and keep myself honest here, in about 40-ish minutes, 45 minutes maybe, because I definitely want to save some time at the end for questions. That's, I think, going to be the most valuable part of this. Right away, I'll disclose, if I can move the slide. Let's see what's going on here. Yeah, I'll disclose that myself and another Hopkins BME colleague, Haichuan Mao, were co-founders of this company that you'll be hearing about today. Now, none of the technologies that we're talking about, none of the products that I'm going to be talking about have been approved for clinical use. So they're in investigational stages only. So there's no conflict in that regard, but it's important to note that I'm a founder and an equity holder, and so is Haichuan in this as we get started. So I want to do three things for you guys today. And we will touch glancingly on that topic of how you evaluate your technologies to see if they're ready. We'll get to it obliquely, and I'll point you to some concrete resources. But I actually want to answer the next question, which is, if you've decided that you've got something that has legs that you think can have an impact on the real world, what do you do about it? Like, what's the first steps? And so I'm going to do that in three discrete steps. The first thing is to really think about why you'd even want to do it. So what's important about biomedical value creation within the academy, within academia, and why us? Why are we academic scientists, engineers, doctors? Why are we the people who should be doing this work? And I'm going to argue for you that we are in an unprecedented time of value creation in biomedicine. And we're so lucky to be working in this time, and we're exactly the right people to be capitalizing on all these discoveries and on the environment that exists right now, both financially and in many other ways, to make it opportune to do venture creation. And so I'll tell you more about that. I will then go to the ground floor and tell you about my own personal experience with this, with a company Chuck mentioned that I helped start at Hopkins called LifeSprout. Out of Hopkins, it's now a freestanding company. And I tell you this not because it's the best example. Certainly there are BME colleagues of mine and broader Hopkins colleagues of mine who've been very successful, more successful than this, whether that's Nick Papadopoulos or Justin Haynes, or there are many other people who could tell you about their particular experiences and could give you case studies. I give you this one because it's my own. It's the one I know best. And so the mistakes along the way are mine. And whatever things we've stumbled on correctly, I think I can speak to with some more authority. And so after giving you a little bit about that, then I want to step back and go to the 30,000 foot view and abstract three principles that I think are really important for biomedical value creation. And that's whether you want to do startups, whether you want to license your technologies. I think these three principles will hold you in good stead. Much of what I'm going to say today is focused on drugs, either small molecule drugs, proteins, biologics, but the same principles I think will apply for devices, for diagnostics, and I will be talking about a medical device as well. So hopefully you'll take the broader principles along with the granular details. So with that said, why now, why us? I mean, I'm making this argument that we're in this unprecedented time. Actually, before I get to Dan Nathans, let me just say this: unprecedented time of biomedical venture creation, value creation. It's a second great wave, I would argue, of biotechnology, really. And for me to argue that, it presupposes that there was a first wave, for me to say that this is even greater and is going to tower over that first wave. So let me say a little bit about that and actually talk about its Hopkins roots. So the first wave of biotechnology, which many of you are familiar with, really had to do with recombinant DNA. That first great age of biomedical innovation, it was from the late 70s to, I'd say the early 2000s. The guy on our right here on the screen is Dan Nathans, who was the director of the genetics department at Hopkins for many, many years. He won the Nobel Prize along with Ham Smith for the discovery of restriction enzymes, which all of us have taken college molecular biology or biochemistry know are the enzymes that allow you to site selectively clip pieces of DNA out of one structure and ultimately put them into something else. That was what recombinant DNA was all about. And it was a watershed because it, one, accelerated the pace at which biomedical discoveries were made. So it accelerated our knowledge, a theme we'll come back to, but it also accelerated our ability to translate that knowledge because it allowed for the production of recombinant proteins and monoclonal antibodies. And in doing so, things like, for example, recombinant insulin, which if you were diabetic back in the pre-recombinant DNA era, you had to purify that protein from organisms, from pancreases of animals. And in the recombinant DNA era, you could actually start making this on demand. And that was true not just for insulin, but for many other proteins of therapeutic import. And then, of course, it also allowed monoclonal antibodies to become a novel modality. So it was really a very important new era that opened up this entire new modality of therapies, proteins, to complement what we'd been doing in medicine for hundreds of years, if not millennia, which were small molecule drugs, things like everything from aspirin to opium. We've known about small molecules for a long time, but since the late 70s, we've now been able to complement that with on-demand protein therapeutics. And not only did that open up a new therapeutic avenue, it created a tremendous amount of value. So companies that are household names now, like Genentech, Biogen, Amgen, these are all great companies that emerged from that first era of biomedical innovation, biotechnology innovation, I should really say. So how do I argue that we're in something even better? Well, and why do I think that's true? I think it's true for several reasons, and I'll give you some examples in a moment. But one of the biggest reasons is that there are so many more therapeutic modalities available to us now than there were in that first era. So things like cell therapies, gene therapies, various kinds of nucleic acid modalities, including modified RNAs that have transformed the landscape in the last 18 months, given the once-in-a-century global pandemic that we're dealing with. And the proliferation of these new modalities, this actually ties to, if I say why now is an exciting time, it ties to the why us. Because in order to use these modalities properly, it places a high premium on biological understanding and on the ability to get these things to the right target cells at the right time. And those are the areas where academic scientists and engineers are really going to excel. They're going to help make these things a reality. I'm just going to give you one example. So Chuck mentioned, I think he mentioned in my background that I was a resident here. So I did my clinical training in plastic surgery and then in craniofacial surgery here. I started in 2011. When I was finishing medical school and I did my pediatrics rotation out at Boston Children's, we had a lot of kids with cystic fibrosis, and we had no therapies for that. I mean, this is one of the premier children's hospitals in the world. This is the late, around when was I doing this? Probably in 2008, 2009. The only thing we could do for these kids with CF is order something called chest PT, which is a euphemistic way of saying that we would have someone come by and then just whack them on the back until they started coughing up a bunch of phlegm. That was a state of the art at the state of the art hospital in 2009, right? Like when I was in medical school, not that long ago. And fast forward only a few years, the first broadly effective therapies for cystic fibrosis became available, and those were small molecules. Similarly, for surgeons like myself, Hep C and other communicable diseases that can be transmitted through blood are of significant concern. We want to always protect ourselves when operating. There's no therapy that was broadly efficacious for Hep C when I started. And again, just a few years later, broadly effective, widely available therapeutics that can cure Hep C in the majority of patients became available. And those are still with modalities from the first age, things like small molecules. Even for advanced melanoma now, things like Keytruda, which is an antibody drug that can harness the power of the immune system to go after metastatic disease, or at least advanced disease, that came on in 2014 as well. Antibodies, small molecules, again, things from the first era, but showing how quickly things have been accelerating over only the last few years. And then it really took off. I think the doors blew open with the advent of these cell and gene therapies, like for childhood leukemia, the CAR T cells, or the first gene therapy for an inherited form of blindness, the first gene therapy approved for anything in the United States, Luxturna, in 2017. Really, in that very short period of time over the last decade, it has been a revolution in biomedicine. There's so many other examples that I can give you today, including the first gene editing clinical trials that are ongoing. So it is a remarkable age. We're lucky to be playing in this age. And I had said a moment ago that that all of these modalities place a real premium on biological understanding. And that calls out to us scientists, physicians, engineers to really build that understanding and aim those tools in the appropriate place. I'll just give you one example again from Hopkins. This is a paper from Bert Vogelstein, Ken Kinsler, Nick Papadopoulos, in which they sequenced the landscape of, I think in this case, this was pancreatic cancer. No, it was colorectal cancer, that's right. And they, these mountains on this landscape are genes that are very commonly mutated, things like P53 or APC or RAS. And then the hills are things that are also often mutated, but less frequently than the really tall peaks here. And you can see it's a pretty diverse landscape. And so if you have a gene therapy that can deliver, say, an that can deliver TP53 in a cancer cell that is missing it, is that going to be enough? Or do you also have to hit some of these driver mutations like RAS? And do you also have to give the tumor suppressor APC? And what about these other hills? What's the right combination? What happens if you get those genes into non-malignant cells? Is there deleterious consequences for those? These kinds of questions are the ones that, again, those of us on the front lines of making these discoveries are the people who can best understand those. And those of us who take care of patients in an academic medical center can also understand how these things are deployed. And of course, for this audience, those of us that are engineers can think about how to effectively deliver those. And that often gets overlooked, but it's absolutely instrumental. There's no point in having, effective mRNA modifications that can lead to protein generation if you can't get them to the right place and expressed in the right cells and in the right time. This example from the right, it's a lipid nanoparticle, the 4-lipid structure that is undergirding mRNA vaccines like from Pfizer and Moderna and others that, again, have taken the world by storm over the last year. Without such a delivery vehicle, those technologies would never have gotten off the ground. And it's, again, engineers, biomedical engineers in particular, who are actually able to make stuff. And that making stuff, whether it's a nanoparticle, whether it is an engineered protein or nucleic acid, that making stuff is going to be instrumental to realizing the value and to capitalizing on the opportunities of this great age of biomedicine that we're lucky enough to live in. So I'm enough cheerleading. I mean, hopefully you guys are convinced that this is something exciting to be doing. I want to transition now and give you, as I mentioned, the example from my own experience of when I was a resident and now a faculty member, how you take a discovery that some colleagues of mine at Hopkins and myself were fortunate enough to make, and how we turned that discovery into something that is both investable and then ultimately we hope can become a series of products that have an impact in the real world for our patients. So I'm going to switch now and go to the ground floor and tell you a little bit about the story behind LifeSprout, this startup that, again, starting way back with the Coulter Grant, we kicked off. So this is our company. It's based here in Baltimore. It's just a few miles from where I'm sitting here on the East Baltimore campus. And we have this mission to develop revolutionary products for tissue replacement and regeneration. And everybody has some grandiose mission statement like this. We were very thoughtful and deliberate about this. So the company really does exist to make products. It's not a discovery company. It's a product development company. And not just any old products. We're not really interested in making Me Too, yet another implant or filler or anything that is already well served in the marketplace. We really want to make things that are quite differentiated and address problems that heretofore have been looming. And the point that we make about tissue replacement and regeneration, we're really thinking about that. So something that can immediately replace tissues, but has a regenerative capacity over time, because that's really where we think the promise of these kinds of technologies is actually going to be realized over the next decade and more. The problem that we started in was one very close to my heart, both as a practicing surgeon, but also as a scientist. So in my BME lab, I run a developmental biology lab focused on skin and soft tissue, both its development and its regeneration. And in my clinical practice, I deal with restoration of soft tissue defects. So soft tissues are things like skin, fat, muscle. They're the kinds of tissues that give our body form and in many cases contribute to function as well. And these tissues are lost in patients many times a year, millions of times a year, from things like congenital malformation, think about like cleft lip patients, for example, to inflammatory diseases like in this young girl with Romberg syndrome, where the soft tissue on the left side of her face has atrophied, leading to this characteristic cul-de-sac appearance, where it's almost like she's suffered a cut on the left side of her face there. From things like that to cancer resection, breast cancer being the signature example, but unfortunately, 1 in 8 women in the United States now get breast cancer and have to have some of the soft tissue of the breast along with the tumor removed, necessitating some level of reconstruction. From everything like that to even simply the process of aging, as the soft tissues of the face and other parts of the body atrophy and descend, it leads to a more hollowed out and skeletonized appearance that many people don't like and want rejuvenation for. So for all of these reasons, soft tissue losses are a ubiquitous clinical problem. And when we were thinking about the antecedent research project for LifeSprout, we realized that our ways of treating them were really suboptimal. We had surgical procedures like fat grafting here or free tissue transfer that could provide natural reconstruction. It's replacing like with like, but it had a significant set of drawbacks, including the need for invasive surgeries, the cost associated with that, and their attendant morbidities, donor site morbidities, unpredictable results, many other problems. People generally did not want to go under the knife to get these procedures. And so what we identified as a critical need, which is the first step in this. I mean, Chuck asked me to tell you guys about how do you know if your technology should be commercialized? Well, the first thing you should think about is, are you actually addressing a problem that matters? Is it something that anybody cares about? And so for us, the need that we clearly identified was that there was no product out there, despite how common these soft tissue losses are, there was no product out there that could immediately restore that missing soft tissue volume while encouraging regeneration over time. So in other words, while behaving like native soft tissue. And that's what we aim to go after. We took a very blue sky approach to this, really went to first principles, which is another thing I would really encourage you guys to think about with your technologies. So we first canvassed the existing product and procedural landscape. So what's already out there to address this problem? Everything from implants, like breast implants for larger volume reconstruction, to the aforementioned surgical procedures. And against those, we elaborated what we thought were the characteristics of an ideal solution. So if we make anything we want, what would it look like? Well, we wanted something, sorry for this typo here, I don't know what exactly happened here, but anyway, we wanted something that was, this is the perils of going from Mac to PC, so forgive me for that. But we wanted something that was made of readily available starting materials that had a long history of safe use in medical devices. We were very deliberate about that because we felt safety is going to be a critical parameter, certainly with regulatory bodies, but for us, we wanted to make sure that whatever we were building was very safe because the existing solutions work reasonably well, despite their limitations. We wanted to be minimally invasive, suitable for these larger three-dimensional defects. And critically, as I mentioned earlier, we really felt that the field was moving toward these more regenerative solutions, cell-based solutions. And so we wanted a capacity for tissue regeneration and cell delivery to be built into the technology. We worked on this in the lab, and I'm skipping through years of work, largely by my colleague and now co-founder of the company, Russ Martin, Xiaowei Li, Brian Cho, who's a resident with me at the time. Many other people contributed to this. And of course, Haichuan Mao, who is the intellectual fountainhead of all of this work, is a colleague of mine in BME. And all of this resulted in a paper we put into Science Translational Medicine that described the first really broadly efficacious, we think, solution to this problem. So a product that can immediately replace soft tissue volume, but then can encourage regeneration over time. And this is how it works. I'm not going to really bore you with the technical details here. This is more of a principles talk, but it's a composite material composed of both nanofibers, in this case polyester nanofibers, coupled to a hyaluronic acid hydrogel. As a consequence of this, it retains this macroporous structure, as is illustrated on this next slide. This is the thing that was really an aha moment for me when I first saw this slide in lab meeting from Russ, where we took native adipose tissue on the left in this electron micrograph and compared it to the structure of the regenerative matrix. That's what we call it. That's a brand name for it in LifeSprout. The term we use for it in technical parlance is just the nanofiber hydrogel composite. But if you look at it, they are really structurally similar. They both have, again, this macropore structure with an underlying fibrillar architecture. The regenerative matrix, it looks essentially identical to living adipose tissue. And that's despite the fact that there are no cellular or animal-derived constituents whatsoever in the regenerative matrix. It's totally a product of synthetic chemistry. And that structure immediately lent us the idea that it should encourage cellular and vascular ingrowth into these spaces, into these lacunae. And that is what we observed. I'm actually going to skip this slide, I'll come back to it. But when we put this material in animal models, as is shown on the right with the composite matrix, I think you can see my pointer here, and compared this to existing hyaluronic acid-based hydrogels, in the hydrogels, there's a relative paucity of cells. But there's some that grow in there because they're pretty dense, but not much. And similarly, relative paucity of blood vessels compared to what we saw in the hydrogel nanofiber composite, where there's a robust cellular and vascular ingrowth. And you can tell this is where our material was because we pseudo-colored the fibers in this case. They're fluorescently labeled, but a much more robust cellular response into the material. And it's completely contained within the material. So as the material is remodeled, the body's own tissues grow in. And you can see this in another way when we injected this into rodents. We implanted the material subcutaneously in the rodents. We followed them with MRI over time. What you see with the LifeSprout materials is that this water bright image, I always get this confused whether it's T1 or T2. I'm married to a radiologist and I still can't remember, but whatever. One of the signals is bright. Surely someone on this call who knows the MRI stuff better than me will know this. But over time, you see that the material has the same attenuation as the surrounding soft tissue. So again, it's because the soft tissue ingrowth and regeneration that we're ultimately seeing over time replaces this water bright material. As opposed to with the leading commercial HA filler, which is water bright in the beginning, just sort of stays that way until it's ultimately resorbed. So it's just like a balloon, it's a space filler. This thing, our thing, occupies the space, but then is ultimately replaced by the body's own tissues. Now that was the science, we published the science. The question is, what do you do with something like that? And this again, I said I would touch on the question obliquely about, is your technology ready for commercialization? Well, I mean, the first question you're gonna have is, what would you do with it? For many technologies, unless it really is a bespoke thing that's meant to solve a single problem, many of the things that we work on as academicians are platforms. They can be used in many different ways. And so one sort of matrix that helped us, one filter is probably a better word for it, that helped us evaluate what we ought to do for it was this one. We were thinking about market size if we wanted to translate it versus the time to realize that value. You know, one thing that I'm really excited about, just and Haichuan is because of our academic interests, is in cell delivery and in cell therapy, broadly speaking. We can come back to that if there's time in the Q&A part. So, you know, this material that encourages cellular ingrowth and encourages vascularization, that remodels and seamlessly integrates with surrounding tissues, in a lot of data that I haven't had time to bore you with, but that excites me. when we know that's true, we could use it to deliver cells. And there's so many people developing cell therapies of various kinds. That's clearly an extremely valuable and attractive opportunity with a huge market size. The challenge is, if as we were starting the company, it's hard to develop in that space. It's long regulatory pathways, actually relatively ill-defined regulatory pathways, because these cell therapies are only coming online now. So that was hard. That's like this corner of the graph. And then on the other side are things that are relatively more prosaic. So we could use this as a wound care dressing, like as a hydrating sort of salve for wounds that people could have for various reasons, either chronic wounds or acute ones. And that's something that we can do easily, relatively easy to translate, very well-defined clinical path, but not so exciting from a market point of view. And so we settled somewhere in the middle with things like a regenerative dermal filler. That was our first decision as a product. And that's the first product that we've actually advanced into clinical trials. So this is what the product looks like. This is the box. It's called Lumina, or at least it's an illustration of what the box looks like. This is the syringe. And it's an in-office injectable filler for age-related volume loss, for aesthetic augmentation. And as I say, this product is not clinically approved. It's being tested now. Our aim is to get a clinical approval for this, hopefully, in the not too distant future. But we chose that for a number of reasons. One, there's a very well-defined regulatory path for that class of products. And the other thing is it's a rapidly growing self-pay market. It's a multi-billion-dollar market. It's growing about 10% a year. And importantly, we felt that we had something differentiating, because we felt that as patients, particularly younger patients, were seeking these aesthetic procedures, they were looking for things that were more durable and also more regenerative, that behaved much more like their own tissues. They wanted to look less done, so to speak, less sort of enhanced and augmented and more natural. So we felt that our material, which feels and looks and performs a lot more like living tissue, could be a solution to that. So we thought we could capture a significant part of this growing self-pay market. In order to develop it, and I'm coming to the end of our part of this, required a lot of stuff, a lot of individual steps, filing IP, raising money. I mean, I mentioned that Chuck was the first 100,000 into this thing through the Coulter Foundation, but subsequently we raised a $6.5 million seed round, and then a north of $25 million A round to help take us through these many steps. So from research to then development and actual GMP manufacturing, ISO testing of these materials to even allow us to put them into clinical trials. And then, of course, earlier this year, despite COVID-related delays that are kind of stretching our clinical timelines, at least we were able to get the first patients dosed with the materials. And I'm condensing years and years worth of work into a few seconds of discussion here, but it really requires a lot of effort. And probably had I known it was this much, I'm not entirely sure I would have done it, but that is another key consideration for you if you're thinking about, does my technology, is it something that I want to commercialize? You also have to wonder, is it something that you personally want to commercialize, or is it better handing it off to somebody who would license it and take it on at a big company if such an offer were forthcoming for you? Oftentimes, we in JHCV actually encourage that. Your odds of success can be higher depending on the type of technology, and you get to do what we as faculty love doing, which is our day job, discovering things, as opposed to perhaps managing some of the minutia of this stuff, which is absolutely critical, but doesn't always play to our academic strengths. You have things like manufacturing scale up, or getting regulatory bodies to sign off. It's not really where we're trained. So this is an important slide, I think, to illustrate some of, only a fraction of, the complexity associated, the long timelines required, and the substantial cash required to make these sorts of things happen. And this is for one discrete product, which is a device, not even a drug, in the context of a larger drug and regenerative medicine opportunity. It really takes a lot of time and money. So I'll summarize the LifeSprout part of the story here, and then I want to move on to kind of general principles. So we like this technology. Obviously, it's ours. We're biased. But we think it is a scalable, off-the-shelf solution to a really important problem, which is soft tissue restoration. And importantly, the same core virtues of this technology in tissue restoration. It's seamless integration. It's very predictable degradation into inert byproducts, because after all, it starts out with fairly inert starting products, and its capacity to encourage cellular and vascular ingrowth in this defined space as it gets resorbed. We think that also has important virtues for regenerative cell therapies to help them become functionally integrated, to help solve the last mile problem. If you're making a beta cell for diabetes, or a cardiomyocyte for heart disease, or a Purkinje cell for neurodegenerative conditions, getting those cells to functionally integrate and survive and contribute to the cure is non-trivial. And we think that that's the direction in which the company is going to go in the years to come. We're really excited about that too. We took a very established systematic path to build value. So as excited as I am about that vision, didn't seem like the place to start because of the challenges in realizing it. So we'll start getting on base with something really concrete and valuable, and then use that as a springboard to these larger opportunities. And as I say, the first programs are now in clinical trials. Let's see how that pans out, and hopefully both the European and US regulatory agencies will be favorably disposed once we get that data in the coming years. And that'll allow us to then fund the really ambitious programs in cell therapy. All right, that was maybe 10 feet or 10 inches above the ground, really a ground level view. And I, again, glossed over a lot of the details, which we can come to in the Q&A, but it gives you a flavor of what it's like if you make a discovery, if you publish a discovery, and you think you want it to get out into the real world, and you want to do something about it. If it's the startup path, that is something about what it takes and what it's like. And I'm going to come now to tell you about more general principles of biomedical value creation. I say value, not venture, because again, you can do this in your own venture, in your own startup, but you can also do this by licensing your technologies to established companies. In either case, there are important principles that I think carry through. I'm just going to highlight 3 of these for you. Okay, and the first one sounds totally obvious, but you'd be surprised how often people don't do this. One of my colleagues at JHTV, Brian Stansky, he has many, many great quotes, but one of his quotes that I love is what the number one reason startups fail is they build something nobody wants. And I might be misquoting him. I think Brian's on this call. He can correct me in the Q&A. But roughly that is what he said. And I think that's really, really true. You got to work on really, really important problems. Now, you can define that various ways. I would say it's major areas of unmet need, major market sizes. I would say it's $1 billion is the starting opening ante. And don't delude yourself and say it's going to be $1 billion because you're going to capture 100% of the market and it's going to grow at 100% a year over the next 5 years. Really, it has to be an established big market, because you could fool yourself, but you won't be able to fool your partners or the investors that are going to provide the critical support needed to actually get this thing out of your academic lab. So you really have to think about a very important problem and a really large market opportunity. And of course, the obvious corollary, make sure you've got a breakthrough solution. If it's another Me Too solution, for a while that can work. So if you have the first modified RNA therapeutic and RNAs are all the rage, then, and you're not Moderna and you're Translate Bio, you can still make it happen. But once you're Translate Bio number 100, in other words, the 100th player in the space, it becomes much, much harder. So try to have a truly breakthrough solution rather than a copycat technology. That's really important. I will also say that this somewhat cuts against the grain of how we think a lot of times as academicians, right? We are rewarded for mastering, in many cases, a relatively narrow field. Like you subspecialize, super subspecialize, and you are the world's expert in that arena. Nobody knows more about this particular domain of whatever area within academia than you do. Well, then you're rewarded. This person, she becomes a full professor at 35 because she's mastered this very narrow area, whereas her colleague who's working on something much broader, it doesn't always lend itself to the kind of linear, predictable academic promotion path. But that's very different in the venture world. It's very different in the commercialization world. You want to think about the biggest, largest opportunities, not the most narrow and focused opportunities. So let me give you a couple of examples. So what's an important problem? CAR T cells. Many of you know what CAR T cells are. This was developed in academia, in a couple of different places. Penn did a lot of the important work. If you look at something like Yescarta, which was sold to Gilead for $12 billion, it's this whole concept that you could engineer T cells to go after tumors, which seems like old hat now, but is only a few years old as a commercially realized product, and in fact inaugurated the era of cell therapies as products. it's transformational, right? It's a hugely important area. It's not extending metastatic cancer by another day or by a week. This is something that can cure disease for patients who heretofore had a death sentence. That is a hugely important problem with a breakthrough solution. Another example, one of my favorite examples from academia, is this one, Luxturna. So this is the first FDA-approved gene therapy for anything. This was developed just 90 miles up the street from us at Children's Hospital of Philadelphia. That's where the foundational technology was developed. And in fact, that's where this company, Spark Therapeutics, was launched. So this went after a rare genetic disease called Leber's congenital amaurosis. It only affects a few thousand patients in the United States, but it leads to profound and early childhood blindness. The children are usually born sighted, but very quickly lose vision due to retinal degeneration that's irreversible. So it's really devastating, you can imagine, for those children and for their parents. And it's a single gene defect. We knew for many years what the gene was that was responsible for this, but we didn't have an effective way of delivering this. These guys figured that out, developed the first AAV gene therapy to deliver the corrective gene to the back of the eye in a selective fashion. And the CHOP actually had the vision to not only do that, not only fund and bankroll the research, but to found the company. And they owned a significant portion of this at the time that Spark was eventually sold for billions of dollars to Roche. I think CHOP owned about 10% of it. So really visionary on many, many levels. But again, a really important problem. This is not a huge market size. It's actually a very rare disease, but a hugely important problem. If your kid is going blind and this is a therapy that can fix it, imagine the morbidity and the cost of lost productivity into the healthcare system of having a person blind throughout his or her life. That is a hugely important problem with a breakthrough solution. Those are the kinds of things. These are, you may not have a gene therapy to cure blindness or make the paralyzed walk. It doesn't have to be that big, but it's the caliber of opportunity that I'm encouraging all of us to be thinking about. I'm going to skip this one. I'll do it in 30 seconds just because I can't help myself. This is one of my favorites. It's a local one. This is our friends Bert Vogelstein, Ken Kinsler, Nick Papadopoulos, who developed this blood test for human cancer. So this is a diagnostic. If the last two were therapeutics, this is a blood test that can detect 8 of the most common human cancers with a specificity of greater than 99%, just from blood, from multi-analyte circulating nucleic acids and proteins in your blood. And it can detect these cancers at a stage where surgery will be curative. If you think about it, the vast majority of effort, including this CAR T thing I told you about a minute ago, is all directed toward metastatic disease, end-stage disease. Whereas if you could detect cancer early on, a surgeon like me or anybody, any surgical oncologist can cure it by just cutting it out. You don't need to go to these exotic therapies. But the problem is, by the time it's detected, it's usually much too late, and those surgical cures are no longer available. So these guys figured out this blood test. They did a clinical trial associated with it, which is ongoing. And they started a company which very fortunately, due to their skill and the great skill of the entrepreneurs that came along to help them do it, got sold to Exact Sciences for $2 billion. And it's my sincere hope for myself and for everybody else on this call that in a few years, their technology is going to lead to a blood test for all of us to see if we have cancer. It's as simple as a blood test for diabetes or blood test for heart disease. It'll really be a landmark, I think, in American medicine. Again, very big problem in this case in the diagnostic space with a breakthrough solution. One other thing to say about this last element about important problems and breakthrough solutions. Another way of looking at it, this came from Peter Thiel's book. Peter Thiel is a venture capitalist. He founded PayPal and a number of other companies. He was an early investor in Tesla and Facebook and many others. He talks about it as 0 to 1 versus 1 to n. The example he gives here is globalization as a classic 1 to n kind of innovation. So think about it. I mentioned radiology earlier. Think about for radiologists who need to read studies, one solution, a 1 to n solution, is something like Nighthawk, which is a service that takes advantage of the time difference in countries with a lower cost of labor. So for example, in India, you may be able to hire US board certified radiologists who will be reading studies in the daytime for them, but overnight for us, and can give you real-time reads in a time where it would be hard to, particularly in smaller hospitals in the United States, to justify having a full-time radiologist on your staff overnight. So it solves a problem. It's definitely an innovation, but it's a one to N innovation. In either case, there's a human being reading the study. It's just depending on where it is. A 0 to 1 innovation, a leapfrog innovation, is eliminating the radiologist altogether. And I mean, I hesitate to say this. I said I'm married to one. I don't want to take food off our table, but it is eventually coming where you're going to have automated image analysis and an algorithm that can, and in fact, these have been demonstrated in the last few years for certain kinds of diagnoses. They can at least match, if not exceed, the performance of board certified human radiologists. So it's remarkable. So that's obviously where things are headed. That is a 0 to 1 type innovation. Those are the ones that, again, people should really be encouraged to pursue. If you're thinking about, is this something worth doing? So another really important one is working with great people. I'd already alluded to the fact that the process of technology translation requires a skill set that we academicians are not always trained to have. It requires a number of skill sets, as a matter of fact, and it's rare, if not unheard of, that one person would have all of these. Whether it's intellectual property, fundraising, regulatory, manufacturing, quality. I mean, there's so many steps along the way that you're going to need to build a great team. So that visionary entrepreneurial leadership that we can provide as the scientists, that is indispensable. I'm not aware of an example, certainly that I've come across at JHTV, where there wasn't a great entrepreneurial leader, a faculty leader who is driving it at the beginning. It's their passion that gets these things excited. It's very hard to outsource that passion credibly and get people to give you money just by getting a hired gun at the early stages. Having said that, visionary entrepreneurial leadership is insufficient. So you got to get great people around you. And it's also a gut check. If you can't get great people to join you, then maybe what you're doing is not that great. It's not that important. And it's another check to answer the original, the ostensible question behind today's talk, which is how do you think about, is my technology ready? Do other people find it exciting? So here's a visionary guy, right? I mean, his paintings have appeared throughout this talk. Let's pick another one. This is a visionary entrepreneurial leader we all know, sadly lost a number of years ago, Steve Jobs. And we all know his story too. He flamed out at Apple when he first founded it, flamed out at Next, his second company. And it was his third act that made him so wildly successful, because he stopped trying to do everything and focused on where he was a genius. And he was a genius in a number of ways, but one of the most important ones was synthesizing the best of technology and turning it into really, really great products that you just couldn't resist having. And I've got maybe 24 of them around my house, and I'm wearing one now and talking to you on another one. So, the guy clearly got it, but he got it because he brought in this guy who's now the CEO, of course, Tim Cook. But at the time, Steve had his third act at Apple, second act at Apple, third act in as CEO of a major company. He didn't run the operations. He had Tim Cook run the operations, a guy who's much more logistically focused, consensus building. And now Tim himself has blossomed. Of course, I don't need to tell you the story of Apple. If you guys have followed the stock over the last 10 years, he's done great stuff. So build great people around you. In my case, I was very lucky at the beginning to work with, and you guys can all be similarly lucky because we have such great faculty colleagues and student colleagues, postdocs. I mean, everybody around this place are so great that, in my case, it was Haichuan Mao who provided the chemistry and chemical engineering, the material science background to realize that vision. If we wanted a regenerative technology with these kinds of properties, and I knew clinically and sort of biologically how such a thing should behave, it was Haichuan and Russ Martin, who's not pictured here, his graduate student at the time, who could actually realize that both operationally by making those molecules, but also thinking about structures that would be that would be biocompatible and realizable. And then this guy, Adam Gridley, who's a great friend to both of us, Haichuan and myself now, who is the executive chairman of our company and had started one of the first aesthetic dermal filler companies and gotten it sold to Mertz Pharma and spent a number of years there as a vice president, was a perfect complement on the business side to the academic energies that Haichuan and myself brought. In fact, I'm about to have dinner with him in half an hour. He's just flown in from Boston, but a great, great person. You have to have great people around you or you're just not going to be successful. And the other thing is, there's so many miserable days being a startup founder. You need other people to buck you up during those those times that are difficult. It's so much more fun to be pursuing these challenging problems with great people around you. So the last one, and this one I really have to credit Mike Weisfeld, who is a legend at this place. He was the former chairman of medicine at Hopkins. He was the first medical director at Johns Hopkins Technology Ventures. He was on the Coulter panel that gave us that first award and became a very close friend and mentor for the company and also for me personally. And I'm so grateful to him. He's the one who told me this and has harped on this for my company. And I pass on this message to everything that I get through the door at JHTV as now myself medical director. And that is get to human as quickly as you can, whether it's a device, whether it's a drug, really getting to first in human proof of concept. That's what really matters. The one step before that is get to a definitive animal model. So something that's going to convince you that the human thing either will or will not work, whether that's non-human primate, whether that's a really well-established translatable rodent model, whatever it is for you, that's the first thing. But the real step function in value comes at first in human. So get there as quickly as you can. Do the regulatory guidance is absolutely critical. It helps you spend the early, most valuable dollars that you're going to get either from translational funding or for your academic grants on things that are going to actually move the ball forward for you, as opposed to doing the wrong animal model, something that is not going to be accepted by FDA, and then you got to go back and do them all over again. So get that guidance early. There's major advantages of being in an academic medical center. The FDA will actually pick up the call. They're very, they love meeting with us, actually. It's really remarkable. And in the pre-COVID era, you just drive down to FDA at Bethesda and you'd have those meetings. You'd have to schedule them, of course, but they're willing to meet with you. And you got to take advantage of that aspect of being in academia. So that's it. I'm going to stop here, and I think I've saved 10 minutes for questions. We're in a great time for biomedical innovation. We're the people, scientists, engineers, physicians who should capitalize on that to do it effectively, work on important stuff, do it with great people, and get to human proof of concept as quickly as you can. If you want to learn more about this, we got a huge team at Johns Hopkins Technology Ventures, ventures.jhu.edu. You can learn a lot more. There's actually something on there called the 7 questions for commercialization. If you're going to disclose a technology to our office, we're going to ask you to answer those 7 questions. They will directly help you answer. Again, the question that Chuck asked me is, how do you think about whether your technology is ready? Go through those 7 questions, and that'll help you figure it out. Let me thank the people who've been instrumental in what I've been able to present to you today. So my colleagues at JHTV, starting with Christy, who's just fantastic, and just such a great leader whose model and whose example I try to follow in my own leadership. Brian, Christy, I mentioned Jorge, Mike, many others. It's really great to work with them. My co-founders in LifeSprout, and then our regulatory consultants too. That's been instrumental. I'll stop there. I'm going to stop sharing screen, and I would be delighted to take any questions.